The notice requirements of the Open Public Meetings Act for this meeting have been met by transmitting the notice of the workshop meeting to the Commissioner's official newspaper, posting the notice where required, and filing a copy of the notice with the Commission clerk. This meeting will be, the Deal Lake Commission meeting will be audio and videotaped and will be shown on APTV, Optimum 77, and throughout most of Monmouth County on FiOS 30. Allenhurst? Asbury Park?
Yes.
Deal? Interlaken? Luck Arbor?
Here.
Neptune? And Ocean?
Here.
Okay.
Okay. Thank everybody for attending. We have a little workshop here, and I welcome the Monmouth University folks, and Jeannie Toller is going to be doing the introductions. Jeannie, go ahead.
Sure. I really feel honored to welcome our special guests and presenter tonight from Coastal Lakes Observing Network and Monmouth University. So first, I'd like to just acknowledge Erin Conlin, who is a citizen scientist coordinator for Monmouth University. She's been training our dedicated water samplers. I know there are some here tonight, so you'll remember Erin, of course, and she's been training our dedicated water samplers all over the lake. She has a BS, a bachelor's degree with a concentration in marine and environmental biology. And tonight's presenter is Dr. Jason Adolph, who founded CLONET. He'll talk to you more about that, the Coastal Lakes Observing Network. Dr. Adolph teaches at Monmouth University with a BS in marine biology, an MS in botany, and a PhD in biological oceanography. We're really grateful that Dr. Adolph has been testing the water on Deal Lake since 2018. And he's going to talk to us tonight about the data he's gathered since that period. So welcome, Dr. Adolph and Erin, and welcome everybody here tonight for attending the workshop.
Thank you. Thanks, Jeannie, Don, rest of the Deal Lake Commission. Actually, I want to talk to you tonight about the data that you've collected. Mm-hmm. We have collected a separate data set, but I'm not going to cover that tonight. Okay, sharing screen worked? Yes?
Yes, we see it.
Okay, so yeah, like you said, Jeannie, I'm talking about CLONET, which is the Coastal Lakes Observing Network that was started through Monmouth University in the Urban Coast Institute of Monmouth University in between 2018 and 2019. We envisioned CLONET as a community-based and participatory research and restoration group. And the big difference there between, if you're familiar with citizen science programs, is that we had always envisioned, and the way it worked out was that the citizens that are part of CLONET actually participate in the data collection and in the research that we're doing in trying to better understand coastal lakes and provide data that's useful towards restoration. It's built on a long history of Monmouth University and Urban Coast Institute and DEP working with coastal lake communities. And what we were going for was to move from a lake by lake solution to being a hub for all coastal lake communities to work together and communicate and understand all the coastal lakes of Monmouth County as a system more than as individual lakes. And we've accomplished a lot towards that goal. And I want to take time here. I don't know how many samplers there are on the call. I see some out there that I recognize. One of the things I regret about these Zoom meetings is that I don't get to see and meet the people that are collecting the data that I'm talking about today. I get emails across my email telling me when a sample has been uploaded to the database. But we/you have really accomplished a lot. There's been over 1,000 samples taken at coastal lakes since May 2019. We've covered all 10 lakes and about 34 different stations around those lakes, meaning some lakes are sampled at more than one location or have been over time. And there's been quite a bit of student participation along the way, which is great. One of the collaborators of CLONET is the New Jersey Department of Environmental Protection, and they have contributed at no charge 701 nutrient analyses from 2019 to 2022, minus 2020, which I'm sure if you're like me, you want to block out of your memory. The lab wasn't open all summer. There were no nutrients done that sample, or that summer. But citizens continued to sample during that summer, I will say, which is very impressive. On the Monmouth University side, so the program I'm not talking about that my lab staff and students have done, we've taken parallel with the citizen measurements, 940 different measurements since May 2019, largely through the efforts of Erin Conlon, who's on the call here and who's wonderful, conducted many trainings of citizens to get them to be able to participate in sampling. And we also use our two data sets to do comparisons of what the citizens are measuring and what we're measuring as a quality control feature. And I've shown that at other meetings. I can talk about that. I'm not going to show any data on that tonight. We've had a lot of student participation from Monmouth University and surrounding high schools. Erin and I have been seemingly forever working on a QAP, which is a Quality Assurance Project Plan, which will kind of make our data more official in the eyes of the state. And we've been working on, one of the main things that I like to do with the data is analyze and visualize it to make sense of it. So as a scientist, I like to understand things through data. And for this specific project, it's how much can I understand about the coastal lakes and what can we understand more about coastal lakes through the data collected by citizens. And there's a data explorer, which I'll talk about in a little bit, on the web that posts all of the data and shows it back in graphical format. We've also hosted 4 public meetings at Monmouth University, which were great. They've all been great, but they were the best, in my opinion, when we got lunch. We haven't been able to do that in a long time because of COVID, but we'll be back. I'm looking forward to that. So again, thank you all, because the data I'm about to get into showing is all stuff that you've collected, and I really do think we've learned something about the lakes, and at the very least, collected data that shows things about the lake that we might have guessed before, but now we have data to understand it on. So the citizen scientists are collecting a relatively simple set of lake indicators, I would call them. And I'm going to go through them quickly here. And although they're simple measurements, when you measure them enough and look at the patterns, you really learn something about an environment. The first is water temperature. Just simply measuring the water temperature of a lake, the ocean, any water body is a fundamental ecological measure. Obviously, it's telling us about the warming and the cooling of the lakes. And some of the things we can interpret from that include the annual cycle of temperature, which is a major habitat parameter for anything that lives in or around the lake. If you're a fisher, you know that. Scientists, ecologists know that. We can also conduct within and between lake comparisons and compare the temperature of our lakes now with historical records that we can find, which starts to let us understand what's happening on a climatological level to the lakes as our climate changes. Conductivity is another measurement, and to me as an oceanographer, I always think of it as salinity. Conductivity increases with salinity, but it really increases with what we call the total dissolved solids. Salt is a dissolved solid. And so within lakes, folks interpret conductivity as a measure of total dissolved solids, and it can mean many different things. But in these coastal lakes, it does tell us a lot about the connectivity of these lakes with the ocean. And that's really something that makes these lakes special. And this is something that makes us special, I'll say, in the eyes of the state and the DEP as well, because we've been included in statewide lake grants because the coastal lakes represent a different type of lake system than you find in much of the state, because of that connection to the ocean. And if you look at historical maps, has anyone ever looked at historical maps of the Deal Asbury Park coastline that show these lakes as inlets, where you can sail right from the ocean into the lake? Really cool stuff. Secchi Depth is another measurement that's made by citizens that tells us, on the face of it, tells us about the water transparency or the clarity. We're basically lowering a disc like this down through the water and recording the depth at which it can no longer be seen. And that's the Secchi Depth. But what that's been used for in the lake science or limnology world for a long time, and I'm talking centuries here, or at least 1 century, is it's a very broad and fundamental trophic status indicator. If we know the Secchi depth of a body of water, we know a lot of other things about the water that would take many other measurements to know, like nutrient levels, for instance, algal biomass, HAB potential. But what researchers have found over the years is that Secchi depth really captures all of that. It's a very simple measurement. pH we had been measuring, but I recently learned that our pH kits aren't that good. So I've been phasing that out, but pH is another habitat parameter that's important to different organisms. Here, you can see where different organisms like to be in terms of pH. I'm not going to talk about that tonight. Then dissolved oxygen, of course, is as important to things that live in the water as oxygen is important to us living above water. Most things that live in the water need oxygen to breathe, although they're doing it through gills or other mechanisms, taking it from the water. And there's a pretty well-defined scale here that we know about, where anything from 0 to 2, and this is milligrams per liter, is bad, and things start to die pretty quickly. And like you and I, if we don't have our oxygen for a few minutes, we know it. It doesn't take a long exposure to anoxic conditions to kill things. 2 to 5 is considered borderline. Some species do okay with it, others don't. 5 to 14 is your normal range, and 15 to 20 is possibly too high. There are some diseases of fish eyes that are associated with overly saturated oxygen in water. So these are the things that we've been measuring. And what I want to do now, I've prepared these dashboard figures of the data. And so I'll use this one to explain it. And what we've got here is, for each parameter, I'm going to go through something that looks like this. I'm going to show you the time series, where the different colored dots are the different lakes measurements of parameters. So this whole slide is about temperature. The next graph over here is it's called a strip plot, and it's all the data I'm going to show you in this plot and the maps is from summer. This is all of the data. So this is the average of all the summers between 2019 and 2021 for which we have data. On these plots, the dots in the background correspond to the individual measurements that have been made on a given lake. The red diamond is the average of all those. So if you want to think about what a lake looks like on average for a summer, you look at the red dots. If you want to know about the range of measurements that people get out of a lake for a given parameter, you look at the spread of different colored dots under those red dots. Can I admit, Mary? I figure
Yes.
if I can see that, everyone else can see it.
Yeah, I just did. Thank you. Sorry.
So for temperature, we found, unsurprisingly, for a temperate lake, that temperature is seasonal. It goes up in the summer and down in the winter. Our range is from about 4 degrees Celsius to about 30 degrees Celsius in here. You can see it repeats year to year. You can also see that this average line going through the data, and you'll see this in all the plots, that some lakes are above that and some lakes are below that. And you can see that here as well. One of the things I've pointed out in the past is that in comparing the contemporary temperature data to historical data, there's not a whole lot of it out there, but there's a Deal Lake study from the DEP in 1978 that recorded maximum temperatures in Deal Lake of about between 20 and 23 degrees. And we regularly each year, as you can see, in not only Deal Lake, but in other lakes, measure temperatures 30 plus degrees Celsius. So there's some evidence that Deal Lake and other coastal lakes have warmed over, see how long has it been since 78? 42 years?
Yeah. Yeah.
And that's consistent with other data around the state showing the effects of climate change on systems. The mean summer temperatures are not uncommonly above 25 degrees Celsius in these lakes, although there's a lot of variance. And the reason that is important, and on the map side of things, so the map, let me explain the map. The size of the dot circle shows how many samples have been taken at that location. And the color of the dot corresponds to this scale down here for the parameter that we're talking about. So this is mean summer temperature. So 25 degrees Celsius in the harmful algal bloom world is kind of a critical number. Above that temperature, harmful algal bloom types of phytoplankton start to have an advantage over other types. And we regularly exceed that in the lakes. The average is actually right on that. So if the average is on that, then we have a lot of time spent above that average. And there are other organisms. I have students in class now that are using the temperature data from CLONET to look at turtle suitability habitat, because they have both temperature dependency and also the sex of their hatchlings depend on the temperature at which they're incubated. And temperature varies between lakes. We've seen that. And you can see some examples here, like Lake Como and Wesley Lake both have a lot of samples taken. And I see you there, Gail and Barbara. There may be other people on the call. I don't know if the folks who do Como are on the call, but those are both very highly sampled lakes. And you can see that their average summer temperatures differ. Wesley Lake is cooler, but Como is warmer. So let's move on to conductivity. Conductivity, again, is our measure of total dissolved solids. Another way to think about saltiness. And in these lakes, again, one of the special properties of coastal lakes is the connection to the ocean, and we have a range of conductivity that you normally wouldn't see in pure freshwater lakes, between 100 and over 6,000 parts per million. And in my estimation, this is due a lot to connection with the oceans in lakes that have it, like Lake Como here, these data points. Although that connection has been changed in Lake Como since we started sampling, and now water only flows one way out of Lake Como. Correct me if I'm wrong, Aaron. I believe that's correct. Wreck Pond, and then parts of Deal Lake in this dark purple. By the way, I had some comments from folks saying that it's hard to tell lakes apart. So I've been looking for a color scale that lets you differentiate 10 different things easily on a graph. It's really hard to find. Other things that I'm not going to go into the data of, but I'd be very, very happy to talk to anyone about it. From the citizen data on conductivity put together with rainfall data that the state collects, we have very strong evidence of the influence of rainfall on lakes. Rainfall is fresh water, it's going to dilute out conductivity, and we see these over different time scales for different lakes, an influence of rainfall and conductivity. So that's telling us something about how rainfall actually interacts with these lakes. And we also have, especially from Wesley Lake, so I'm glad you're here, Gail and Barbara, we have really good evidence that in winter, not frozen, but rainy periods, that the conductivity of Wesley Lake goes way up. I'll show it to you on the Shiny app later. And the only thing I can think that could cause that, and it's consistent with the weather patterns of the time period, is runoff of road salt into Wesley Lake. And I have other students this semester doing an experiment where they took water from Wesley Lake and added salts to it to see what happened. And it killed everything, basically. Then again, looking at the map and looking at the means, there's some variability. You can see places like Deal Lake. There are parts of Deal Lake that are higher conductivity, parts that are lower. Lake Como is high. Silver Lake here, Sylvan and Wreck Pond are the yellow, very hard to see points here. There aren't very many of them with the higher conductivity, like down here. Como has more salt near the ocean, less salt here. And then in Deal Lake, you can see the saltier stations in the lower, more eastern part of the lake compared to up here in the higher parts. Okay, dissolved oxygen. Dissolved oxygen is another seasonal kind of parameter like temperature. You can see that but it's opposite. If you notice, where temperature is high, oxygen is low. And that's a physical property of how oxygen dissolves in water. Warmer water holds less oxygen. That's just physical chemistry. So you can see we have the seasonal pattern on average of oxygen. And we have to keep in mind that in terms of hypoxia and anoxia events that will kill things, that these lakes are going to be more susceptible to that the more warm they become each summer. And that makes sense. The other thing to note about the oxygen measurements, and we have a lot of them now, is that if you recall, I said that between 2 and 5 was borderline for a lot of things. And while our averages are well above 5, there are many measurements shown by the colored dots in the background in many of these lakes that are well below 5 and even down as far as 2. I don't think we've ever measured pure anoxia or lack of oxygen in the lakes. And that's not great. So when we think about nutrients in lakes, what well-oxygenated lakes do is they hold the nutrients in the sediments, keep them out of the water column. Once those sediments go anoxic, or what we call reducing conditions, those nutrients will be released into the water column. And so if you think about nutrients only coming to the lakes from the watershed and the lawns and fertilizer and the street runoff, we also have to consider that there's a bank. There is a bank of nutrients in the sediments of these lakes, and what pulls nutrients out of that bank account is low oxygen. What we don't know is what happens at night. All of our oxygen measurements are made during the day, citizen and lab alike. And that's unfair because during the day, the lakes are photosynthesizing, or more appropriately, the phytoplankton in the lakes are photosynthesizing. And that's going to increase the oxygen concentrations in the lakes. What we need to do, and I have a student doing this summer, we're going to deploy some 24-hour sensors in the lakes to measure oxygen as it goes up and down over the course of a day-night cycle. And Dr. Souza can correct me, but I don't know if that data exists for lakes yet. And then Secchi Depth. Here's the time series of Secchi Depths. You can see that the average line runs about 1.5 feet in depth. With variation that takes it to less than a foot to about 4 feet in depth. And again, Secchi Depth is a key and broad indicator of trophic status measured in water bodies worldwide. And our values all over the lakes, although they vary by lake, they're all on the very, very low end of the scale, which means that our lakes are very eutrophic, if not hypertrophic, So if you're classifying lakes by that measure, our lakes, unfortunately, would fall into the lowest of those categories. And although I'm not showing it, that is backed up. Actually, I'm going to show a little bit of it after this. That was backed up by other measures that we've made and the DEP has made. Again, on the map, you can see the different lakes. Average summer Secchi depth varies by location. So that's the run through our data. I know it's a lot. Now I want to take our heads, open up the view a little bit and look at the DEP's data, where we can compare a coastal lake, which happens to be Deal Lake that they've sampled, to the other lakes that DEP is looking at closely because of water quality impairments, including harmful algal blooms. So that's Greenwood Lake, Lake Hopatcong, which you may recall in 2019 had severe harmful algal blooms, Manasquan Reservoir, which is a drinking water reservoir, and Swartzwood Lake. So this graph on the far left is Secchi depth, and here's Deal Lake. Now these are DEP measurements, not ours, but they come out about the same as we're getting, in the 1 to 2 foot range. Then you have Greenwood with an average closer to 2, closer to 4 in Hopatcong, Manasquan Reservoir, and Swartzwood, all much deeper Secchi depths. So if you look at the nutrient measurements, so this is total nitrogen and total phosphorus, which are two key nutrients associated with eutrophication of lakes, you can see that they show an opposite trend to the Secchi depth measurements. You have high nutrients in Deal Lake, and that's true of other coastal lakes for which we have measurements, and declining nutrients across the other lakes that also have clearer water. That's what I mean by Secchi depth as an indicator. We can look at the Secchi depth and guess what the nutrient levels are pretty accurately. And this is important. I think this is important to put coastal lakes, including Deal Lake, in the context of other lakes in the state that the state has its eyes on. And I would argue that in terms of water quality, the coastal lakes need the most attention compared to these other lakes. They're in worse shape. So in summary, the data set the citizens have collected and we've analyzed and looked at does a really good job of showing the impaired condition of the lakes. And in my estimation, provides a really good argument for investment in the restoration. What I'd like to see happen is for CLONET citizen monitoring and lab monitoring to continue through these restoration efforts. And I know that there are a lot of restoration efforts in the hatching, in plans by different lake commissions, including Deal Lake and Wesley Lake and Sunset Lake in the Asbury Park area. I think what we have is a mechanism to show that we're in the game and that we can measure the improvements that come from that. So I want to say something really quick about how to get involved in CLONET. If there's anyone on the call who would like to get involved or isn't involved yet, contact Erin. She's right there, and that's her email, and she does the training and the retraining. She works with the data, she troubleshoots for you, and she supplies supplies and picks up the waste if needed. We provide the equipment, we provide the training, and we provide the database for you to report your data so it doesn't just disappear on crumpled pieces of paper in someone's office. The reason we have over 1,000 samples the way we do is because of this database organizing things. Getting involved is not difficult. It takes a group leader. This is really why I put this here. To keep a program like this going, is not only local residents, but getting schools, summer camps, and scout troops involved. that will keep the sampling going. We have new equipment. So in addition to the parameters that I showed you, we have through the Watershed Institute and through the New Jersey DP and through the Plangier Family Foundation, HAB monitors. So this is what the state uses to monitor water bodies for harmful algal bloom abundance, and we'll be handing out and training folks to use these this season. This will allow folks to directly interact with the state in reporting HABs or pending HABs, in addition to giving us a better understanding of HABs in the lakes. So at this point, I am interested in hearing either now or you can email me, you know, how do you see these data best being used to help get all these restoration plans and goals over the finish line? I have ideas, and they go like this. If you're writing a proposal, you're trying to convince someone to give you money to do a restoration project. One, the CLONET project demonstrates a community that's engaged in lake stewardship above and beyond the other activities in which you're involved. It's another feather in your cap to make somebody want to fund you to improve something. Two, it provides a baseline of data for conditions currently and in the past, and mechanism to track changes following actions that may be taken. You can see the results of water quality improvement plans that are put into place. And two, it promotes ongoing education of community members about lake stewardship and directly involves people. We are submitting a grant through the Urban Coast Institute to the National Fish and Wildlife Federation to build on CLONET and move it from a monitoring posture or stance to also include restoration planning using the data we've collected. Tom Harrington and I submitted that proposal today, actually. And I will definitely keep you updated on what happens with that. But I think that's where we're ready to go with this project and this this program, moving it towards supporting restoration in addition to just monitoring conditions.
Wonderful.
Okay, so that's the end of my talk. I want to thank the Plangier Family Foundation for funding the CLONET project for 4 years now. Again, all the citizen scientists, can't thank you enough. I want to give you lunch, and I will again. And our DEP collaborators, and thanks to you all for your attention. And I put a little guy doing a Secchi cast to this old Deal Lake picture here. And I'll be happy to take any questions, although I probably.
So
Hey, Jason.
if I might just interject for a moment, if you'd like to ask a question, just raise your hand. And when you're called on, would you please state your name, your address, and then your question?
Steve Souza, I think, had a question.
Yeah, Jason, thank you very much. I mean, you know, this data is definitely invaluable for us. It's just, you know, it's the type of information that we need to really track the status of the lake. And as you pointed out, we have used it already to our advantage to demonstrate engagement of the community. One thing, you know, I just want to, for your benefit and maybe for some other people that might be interested, I'm actually going to be putting together an urban lake management session for this year's North American Lake Management Society conference. I mean, unfortunately, it is in person and it's out in Minnesota, but the purpose of me bringing this up is your point about the fact that urban lakes really require a lot of attention. And I think there's, an increasing, let's say watershed moment, no pun intended, across the country, recognizing the value of urban lakes. But it's something that I think New Jersey DEP really needs to set us aside from, the Lake Apatkongs and the Greenwood Lakes, because, we actually service a much more diverse community. A lot of urban lakes are servicing, overburdened communities. And unlike these big recreational water bodies, we haven't been getting the attention, and the funding that's really needed given the, the magnitude of the problems that we're facing. So thanks very much for, what you're doing, because it is a very valuable service.
Thank you, Steve. I agree with you completely. And I'll take this opportunity to mention that Erin, DEP collaborators, and I just got a paper published in a journal called Urban Naturalists about Deal Lake, 2017-2018 data. And that should be coming out in, who knows, academic journals. It could be like 6 months. But a lot of the arguments you said, Steve, I made in the introduction of that paper. Bob J., I see your hand.
Hey, Jason, great presentation. Just fascinating to look at this data. The things we suspect and all know that the lake needs help, but to see the metrics behind it is phenomenal. I mean, I have so many questions. I'll just ask one right now. On the conductivity slider, when talking about salinity, for Deal Lake, it was obvious by the flume it was a little higher. We would expect that. But it looked like up near the golf course, like the Allenhurst sort of park area there, was higher in salinity. I'm just wondering how that can be. Is that runoff? Is that salt? I can imagine why that area would have a higher salt content.
So I'm looking back at that, and you're talking about the arm of the lake that extends directly north.
Yep, right before it turns.
Yeah, I had seen that, and without running out there and checking it again, I don't have a great explanation for that unless there's a movement of salt in that direction from what comes in at the flume. It could be runoff, and it could be something else causing that conductivity besides ocean salt in that area, like you suggested. It would definitely be something to look further into.
Yeah, I don't get into too far into the weeds here, all right? The data is great, and there's so much we can do with it. Really appreciate all you're all doing.
Yeah.
So I think there's a question from Jim Veach. I can't see it up anymore, but Jim, do you want to ask it orally or?
Oh, yes, thank you. Thank you, Jean. Yeah, I just wanted to address the professor. I appreciated that presentation very much. My question has to do with infill and sedimentation. I'm assuming that as the lakes grow more shallow, all these problems get worse, salinity, et cetera. And I'm wondering if you did any work along the lines of setting a baseline for the depth of these lakes as you were doing these other tests.
That's so the first answer is no, we haven't. The second answer would be that I'm with you, and a lot of people, I think, intuitively, and even in the research literature, you read that dredging lakes like this is a solution to eutrophication sometimes, if the lakes are small enough where dredging is feasible. And I think coastal lakes are, even Deal Lake. In other words, the papers that I've read about lake restoration, maybe not all of them, but the ones I remember, because I want to remember them, say that dredging is a good option. It's just not practical for most lakes. You can't dredge Lake Hopatcong. You can't, but Deal Lakes you can. I think dredging is one of those restoration ideas that's going to be really important to consider and evaluate moving forward. I think that's one of those restoration ideas that we have to move from intuiting and thinking about it to really taking the steps to see if it's worth doing. I will say that in Deal Lake, in the summers, when it gets hot and it gets heavy in there, we see more phosphate in the water column than there was. The phosphate actually increases over time in the water column without rain, which has to be coming from the sediments. So there's got to be a ton of phosphorus locked up in the sediments that when summertime conditions set into these lakes, it seems to me that the sediments just start pouring phosphorus up into the water column. And so you can see how that causes a kind of a positive feedback cycle between the growth of algae that use phosphorus and nitrogen. The more they grow, the more phosphorus comes out of the sediments because now they're dark and reducing. And it just helps establish these harmful algal blooms that happen in the summer.
Thank you.
Jason, do you see the other questions on the chat by any chance?
I do not, and I'm close to running into your meeting time.
Well, let me give one. Kathleen Mana said, Jason, where can we find the historic maps of the lakes?
Rutgers has a wonderful, I've downloaded some, but Rutgers has a wonderful archival map server for New Jersey. And I don't know what the link is because I usually just Google it. If you Google Rutgers archival maps, you'll probably come up with it, and you'll get a list, and you'll see all these old maps back to the 1800s of Deal Lake, the Long Branch.
I saw one at Deal Lake with no bulkheading down by the ocean. It had to be the mid 1800s when it was open to the ocean. It was pretty cool. Just saw it 2 weeks ago actually.
Yeah, they're really cool. I couldn't find a good. Well, I couldn't print one out to put on the slide. Yeah. Otherwise, I would.
They're long, elongated maps.
Right.
Photos.
Right.
Yeah.
Yep.
But I have a contact at Rutgers that's doing a landscaping study on the lakes, actually Deal Lake, and I have his contact information, so I can grab one of those, maybe get it on our website. We can do that.
I don't see anybody else. Any other questions? And again, I know we're all very grateful to you, Dr. Adolph, and to Erin. Before the meeting started, Erin told us that we do need 2 new volunteers to be citizen scientists. So you can reach out to her or to the DLA Commission. Wow, whoever that is, if you don't.
One second. Okay, Jeannie, go ahead. Jeannie. Got to unmute, Jeannie.
Somebody muted me. Okay.
I muted everybody.
All right. So Erin told us just before the workshop started, that we are in need of 2 citizen scientists for Deal Lake right now. So if you're interested, you can get in touch with the Deal Lake Commission and we'll put you in touch with Erin so you can get trained. Or if you happen to take down her email address, she put it up earlier. So just to let everybody know that we are looking for 2 citizen scientists to get trained.
I want to thank him too. And some of the observations I made, 25 degrees Celsius, that's 77 degrees F. That's hot. Pool temperature, right? 30 degrees C is 86 degrees Fahrenheit. And I've been fishing in there and I'll see 82, 83 once in a while. So these numbers are not unrealistic. They're good. It's good data of what I can see in Deal Lake. So you guys did a stellar job. And the nutrient loads, we know we got to get them down. And that's some of the projects we're working on with the treatment devices and some of the proposals we have on the table now. And we use you guys, the references to the CloudNet program to gather funds for Sunset, Wesley, and Deal Lake. And so keep your fingers crossed, maybe we'll get another grant. And they are reviewing that grant application because we're getting questions on it. So that's really great news. And CloudNet guys, Monmouth University definitely helped out. And you guys are considered a partner to us. So we thank you. We thank you.
Thank you so much for coming tonight.
Thank you, Jeannie and Don and everybody.
Yeah, and we got another grant we just saw, and the separate lakes are going to go. It's a $10 million grant that we worked on hard to get funding for, and not just for Deal Lake, but it's all lakes in New Jersey. So Yeah, I know Sunset, Wesley, and Deal Lake are going to go after that. We're going to talk about that in a minute, but and Claudette's going to be heavily involved with that. So again, thanks a lot.